be asking yourself, what purpose do sharks have in directing biomass transfer within an ecosystem? And if you're not, well, you should be. You see, ecosystems have many interactions between the different trophic levels. In documentaries, you might only see this shark eating this fish or this bird falling out of this nest. But in reality, it's far more complex. It seems rather simple. One thing eats another. But let's say I'm in the ocean and I eat a kilo of fish. I don't gain a kilo in body mass. I'll poop out most of that weight and I'll pee and sweat out other waste products, all of which can get used by bacteria, filter feeders and fish, and as long as I'm living and respiring and don't get eaten by a shark, I'm going to be losing 300 grams a day of carbon in the form of carbon dioxide, which will be photosynthesized by plants. After all this, I probably haven't gained a single gram. The point here is that carbon and the biomass associated with it goes through convoluted pathways that don't always involve upward steps in trophic levels. You may only observe the initial surface level consumption of one trophic level by another, and rightly, you would notice that each level decreases in biomass. But this mass isn't destroyed, it is merely lost to the lower trophic levels. Like in my case, a victim of the inefficient transfer of energy up the food chain. What I've just outlined is the law underpinning this form of ecosystem study, the law of the conservation of mass, meaning that any carbon entering an organism or an ecosystem must equal the carbon coming out. And if it seems to disappear, it just means you're not looking hard enough. Let's look at a conceptual example of this and get back to sharks. Ecosystems are sustained by the primary producers. Carbon is fixed using photosynthesis, and this energy and mass flows up to the higher trophic levels. But this is a two-way street. Just as prey exert a control on predator biomass and distribution, predators exert a control on prey, sometimes with ecosystem-wide effects on carbon and energy flows. Sharks alter ecosystems through direct predation and risk effects, and their removal can result in trophic cascades. The direct predation effects of sharks can be quite simply put. Sharks feed on mesopredators, taking away from their biomass and turning it into shark biomass. This leaves less mesopredators to consume their prey, such as herbivorous fish, resulting in an increase of their biomass. In temperate marine systems, it has been observed that when apex predator sharks are removed, this causes the effects to invert. This is called mesopredator release. More biomass stays in the mesopredator trophic level as less is consumed by our apex predator, meaning that the predation pressure on herbivorous fish starts to rise, causing their biomass to sink. This is a trophic cascade, but this is very context dependent, and that example was too easy for you anyways. So let's look at some examples where there is no cascade. As we established, sharks remove individuals of a mesopredator species, but in some, let's say resource-limited cases, all this would do is remove a direct competitor for other members of that species, giving them access to more resources, resulting in compensatory reproduction and growth. Essentially, we could see a lower abundance but larger individual size, giving no net change in the population biomass, and thus no cascade if sharks are there or not. The other point covers coral reefs, where the incredible diversity means that no one apex predator and prey interaction is strong enough to overcome the diversity of interactions in the system and cause a cascade. So the removal of an apex shark species could have little effect, as another would just take its place, benefiting from the reduced competition and increased food availability. Non-consumptive risk effects are arguably more important. This is where the presence of the shark affects the distribution of its prey as it tries to avoid being eaten. But how does risk affect the food web and carbon movement? Well, the distribution and foraging behaviour of dugongs and sea turtles in Shark Bay, Western Australia, is driven by predation risk of tiger sharks. These mega grazers end up avoiding risky habitats, allowing dense seagrass beds to form there. Invertebrate and fish communities are linked to seagrass biomass. Thus, this top-down indirect effect facilitates the diversion of carbon produced in the seagrass beds to go to benthic and epibenthic communities that create the basis of a productive ecosystem and can support hundreds of species of fish and other invertebrates. This allows the carbon to be converted to large amounts of biomass across many phyla, rather than funneling it into a few mega grazers. Now, I hope you learned something from that quick look at carbon flows within ecosystems. But how can we put some hard numbers to this and get a complete picture of the mass balance within an ecosystem from the bottom producers to the top predators? This is where EcoPath comes in. It is an end-to-end ecosystem model that gives a snapshot in time of the mass balance within the system by incorporating everything from the lowest trophic levels like phytoplankton and zooplankton to the higher trophic levels like large fish or us humans. It uses these 6 simple parameters for every biological group in the system. And with these parameters and principles of mass balance, we can equate the production and losses within an ecosystem to learn about the biomass caught at every step of the food web. With the help of some friends, you can collect all the data you need to feed this ecosystem model. And that's exactly what we did at the estuarine ecosystem of Smith's Lake. Being an intermittently closed and open lake lagoon, Smith's Lake is ideal for this form of ecosystem study as it is a fairly closed food web with little migration in or out. The lake has been closed since October of 2018, and due to fishing, the large fish and benthic fish groups have been mostly removed, and there were also less birds. We expected the loss of this predator to result in a trophic cascade, where their prey, zooplanktivorous fish like hardyheads, would grow in abundance after being released from predation, thus causing a decline in zooplankton biomass. But this was not the case. The small fish biomass was still declining, as in previous years. Phytoplankton was blooming. Zooplankton was up from 2019, and jellyfish biomass was at an all-time high. But why was this? To find out, we have to focus on exactly these 4 groups and let the others spectate. Reduced river flows can increase the salinity and temperature of a closed estuary as the water evaporates, leaving saltier, shallower water and reducing dissolved oxygen as the conditions deteriorate. This is exactly what happened to Smith's Lake with a severe lack of rain from November 2019 to January this year. These hypoxic conditions are most likely what stunted the growth of the rest of the ecosystem while promoting the spike in jellyfish biomass. With such a large presence of jellyfish, 80% of which were the carnivorous Catastylus, the small fish had a very poor chance of competing for their shared food source, zooplankton. The EcoPath model shows that 96% of the zooplankton biomass was being consumed by jellyfish, leaving us no longer surprised by the stunted growth of our small fish, despite their predatory release. The actual zooplankton biomass we found in Smith's Lake was 0.73 grams per metre squared, and the calculated biomass, or the amount needed to support all the zooplankton consumers, exceeded this by a factor of 15 times, and that was after we factored out the photosynthetic jellyfish. This tells us that the drought-induced jellyfish bloom went to the point where it was likely exhausting the ecosystem's zooplankton resource at the current level of productivity and exceeding its carrying capacity. You may already be realizing that this system could be on the brink of a jellyfish collapse, but this might not happen as quick as you think. You see here, between the summer drought and the spring sampling, the rainfall around Smith's Lake easily exceeded the monthly averages for February and March. This would have filled up the lake, reducing the salinity, and likely brought a flux of nutrients from the catchment. This might be what gave us our record high phytoplankton levels that actually exceeded the calculated phytoplankton biomass needed to support the system by more than double. This carbon then fed into the zooplankton biomass to sustain the high demand, at least for a bit. And that could be why we see the elevated zooplankton biomass this year. From all this, we discovered that our expected trophic cascade was in a way quenched by this bloom and diversion of carbon to the jellyfish, all driven by climatic variability. So there you have it. We went to Smith's Lake and it gave us a challenge. But after some prodding beneath the surface and the help of our EcoPath model, we uncovered the secrets behind its mass balance for this snapshot in time.